welcome to our histology presentation about the major salivary glands. Development of the major salivary glands, the parotid, sublingual, and submandibular, starts around 4 to 12 weeks. The secretory end pieces, also known as acini, gain maturity during the last 2 months of gestation and continue to grow postnatally up to 2 years. Its development is divided into 6 stages. First stage is the bud formation. Here we have the buccal epithelium in dark blue and ectomesenchyme cells represented in yellow. The underlying ectomesenchyme induces proliferation in the epithelial cells, resulting in tissue thickening and formation of an epithelial bud. Next, we have the cord growth stage, where a solid cord of cells is formed through cell proliferation and condensation, marked as a dark red. Stage 3 is branching of cords. Clefting in distal buds initiates the branching process, epithelial proliferation then follows, and cord differentiates rapidly. Stage 4 is the lobule formation through repetitive branching of the epithelial cord. This continues at the terminal portions, forming an extensive tree-like system of bulbs. Canalization of cords is the 5th stage and happens around 6 months for all 3 major salivary glands. Lumen formation works from the outside, starting from the excretory duct, working its way to the terminal branches. The final stage is cytodifferentiation. Cellular activity shifts to the terminal bulbs. Stem cells in the bulb region undergo self-proliferation, then differentiate into acinar or ductal cells. Simultaneously, myoepithelial cells arise from the epithelial stem cells.
Salivary glands are classified based on their size, major, which we are focusing on this video, and minor. Both types have similar histological features, and their differences are based on encapsulation, numbers, size, and their length of ducts. All major salivary glands are divided into divisions called lobes, and then further subdivision is called lobules, each of which is separated by a connective tissue called septum. Salivary glands comprise of a series of secretory end piece or acini. They are connected to the oral cavity by a system of ducts and then myoepithelial cells. Now we will talk about the acini. Acini are epithelial cells that produce saliva. They consist of secretory cells arranged in a roughly spherical configuration around the central lumen or cavity. And there are 2 main types, serous acini and mucous acini, which will be discussed later in this video. Moving forward, we will be discussing the ductal system. Salivary glands are exocrine glands, meaning they have ducts to secrete substances. These ducts progressively grow larger around the inner parts, from the inner parts to the outer parts, and they may be intralobular or interlobular ducts. Intralobular ducts are composed of 2 subdivisions, intercalated and striated. Intercalated duct is attached to the sinus and they are smaller. Striated ducts are connected to the intercalated duct and they serve as a passageway between the intralobular duct and interlobular duct. Lastly, excretory ducts are interlobular because they are the final part located at the septum. This is where the saliva is on its way exiting to the oral cavity. If you combine the ductal system and the acini, you could picture it as a stalk attached to the grapes.
Corporate needs you to find the differences between this picture and this picture. Intel has told us there were at least 7.
Okay, I already see one. Give them. Okay.
They're the same picture.
Finally, we are going to talk about the myoepithelial cells. They are the crucial contractile cells around the acinis. If these cells contract, the acinis and its contents are squeezed out, expelling the saliva out of the lumen and into the intercalated ducts. They are not present along the striated duct. It is like an octopus squeezing out their ink to confuse the predators as they escape.
The parotid gland is the largest of the salivary glands, and it is responsible for producing 25% of saliva in the body. The majority of parotid gland is made of serous acini, which are responsible for the secretion of thin watery fluid that is rich in enzyme. These enzymes contribute to the process of food being broken down in the mouth. The second largest gland is the mandibular gland, which is responsible for producing 60% of the saliva. The submandibular gland is predominantly made up of serous acini, but it is also contains a small number of mucous acini. The mucous acini produce a more viscous fluid that acts as a lubricant for the mouth, while the serous acini produce a fluid that is transparent and include enzymes. The sublingual gland is the least significant of the 3 glands and only producing 5% of saliva, primarily made of mucous acini with a smattering of serous acini, drawn in for good measure.
Here is our list of references. Please like, subscribe, and feel free to comment any questions that you may have. Thank you for your time and keep flossing like a boss, you dental hygienist.